The Lion hypothesis states that inactivation of X chromosomes will happen randomly in every single cell of an embryo during development. Cells present in developed mammalian bodies only express the genes from one X chromosome. For that reason, during development, all cells in the embryo will randomly choose one X chromosome to leave activated. Each one of the other X chromosomes will then be inactivated by condensing and creating what's called a Barr body. However, when the subject of study is a heterozygous female for the trait being analyzed, things can get a little more confusing. Usually, for the animals as a whole, the expression of the dominant allele in some cells will override the expression of the recessive allele from other cells. But that is not always the case. This can be seen on the coloring of tortoiseshell cats. For example, in these cats, the color of their fur is an X-linked trait. The dominant allele gives black fur and the recessive one yellow. However, what happens when a female has one of each allele as their genotype? In this case, when each hair cell expresses their gene for hair color, they will only show the allele present on the activated X chromosome of that specific cell. That's why the tortoiseshell cats have some spots of black fur and some of yellow fur over their body, and that's also why that coloring only happens in females. Let's see what happens if we add another trait into the mix, their hair length, which is an autosomal trait. The allele for long hair is the dominant one. Let's imagine this cross, a yellow short-haired female crossed to a black male heterozygous for long hair. Using this branch diagram, we can see that the ratio of phenotypes for this cross would be 1/4 female with tortoiseshell long hair, 1/4 female with tortoiseshell short hair, 1/4 male with yellow long hair, and 1/4 male with yellow short hair. Lastly, let's talk about what happens when we clone a tortoiseshell cat. Cloning requires the transfer of the genetic material in the nucleus of the tortoiseshell cat to an enucleated egg. That means that the two X chromosomes go to the egg, one activated, the other one as the Barr body. Since the original donor cat has already had its X inactivation embryo stage, the new cat being born won't go through it. The embryo can already tell that there is only one X activated, and so all the cells that come from the zygote will have the same X chromosome as the Barr body. In practice, what this means is that all the hair cells in the cloned cat will express the same X chromosome, and so it will lose its black and yellow spots. The resulting cat will have the genotype of a tortoiseshell cat, but the phenotype of a fully black or fully yellow cat.